So I'm a PhD scholar in infectious disease program of College of Veterinary Medicine, Mississippi State University. I primarily work with Dr. Hansen, Dr. Weiss, Dr. Griffin, Dr. Perkins, and Dr. Walbiser. So today's topic for the discussion is epidemiology and phylogeny of Ictalurid herpes virus 1. So, as you all know, channel catfish is the principal food fish species cultured in USA, which is mainly spread across Mississippi, Arkansas, Alabama, and Texas. So, when we look at the industry, it's established during 1950s. Later on, during 1980s, it started the intensification. Currently, it's spread over 44,600 water acres with around a $380 million industry. So when we look at the aquatic ecosystem, compared to the terrestrial ecosystem, as you can see, our fish constantly interact with other components of the system, and the interaction is happening at the multiple levels, at multiple dimensions, and all the factors simultaneously interact in. And finally, if any of the factors goes wrong, one day the farmer will show, hey, we have some disease. So it could be due to any number of reasons. So this is a particular challenge as fish virologists or as fish microbiologists. This is what we face. Now, coming to our today's topic, as you all know, the channel catfish virus, which predominantly infects the fingerlings, which is almost less than 1 year old. And the outbreak can reach up to 100% if it's a really bad outbreak. And from the earlier reports, we also know that there is vertical transmission as well as horizontal transmission. And as you can see, the clinical signs with the distended abdomen and hemorrhagic areas on the fin and the ventrum. And it is also known that environmental factors really play a critical role in the disease progression, particularly water temperature. So we usually see more outbreaks of greater than 28 degrees Celsius. So when we look at the classification of alloherpes viruses, we had two categories mainly, that is ICHV1 and ICHV2, which affects the black bullhead. Now, in my work, we have identified a new one, which is called blue catfish alloherpes virus. We'll be talking about that in the talk later. So as a foundation for my work, Dr. Hansen has performed an RFLP analysis on different isolates collected over the time. So it is evident from the picture that these are basically two groups. So RFLP A group, RFLP B group, which is similar to ICHV1A, and there is a distinct isolates present. So this is the isolate later on. In my work, we have identified as blue catfish virus, or we call it as blue catfish virus, blue catfish alloherpes virus. So considering this, my objectives of the studies are basically assess the susceptibility of channel and hybrid catfish to ICHV1 RFLP groups of CCV. Then what is the prevalence of these RFLP groups in our hatchery fry? And how about a genome survey of all the isolates? And we would like to know, is there any significant difference between RFLP A group and B group? So coming to the first objectives, so susceptibility of channel and hybrid catfish to ICHV1. So from the earlier reports, it's known that channel and hybrid show a differential susceptibility. In general, blue catfishes are more resistant, and hybrids show a increased susceptibility towards the ICHV1. Now, in order to test our new, is there any effect of these two groups on channels and hybrid, we designed a study in which we selected 3 isolates from RFLP A group and 3 isolates from RFLP B group. And we exposed the channel and hybrids to this isolates. So we used 3 × 10⁴ PFU per mil dosage with the 4 replicates and 20 fish per tank. So as a challenge model, we basically kept water depth 3 inch, which is enough for the fish to float with the 4.4 liter water. Then we added the virus and we allowed to stay there for 1 hour. Later on, flow through system has been switched on with aeration, and they allowed to stay for overnight. Next day morning, we replaced the standpipe, and normal water depth was restored. And we fed them 2 times in a day, and mortality has been checked 2 times in a day. So from the results, you can see when we compare the differential susceptibility of channel and hybrid towards 2 different RFLP groups, there is no significant difference between channel and hybrid against 2 RFLP groups. Now, if we compare the RFLP group A and B, again, there is no significant difference between RFLP group A and RFLP group in terms of the susceptibility towards channels and hybrids. So now we have identified a new virus, which is called blue catfish alloherpes virus. So we wanted to test that. So in order to do that, we selected again catfish, channel catfish, hybrid, and blue catfish. And we determined the LD50 dosage, and we got a same dosage as of our type strain, which is ATCC O-borne type strain. So rest of the challenge model is same. We did exactly same what we did for other RFLP groups. Now, this is a really interesting results where the blue catfish shows 50% or 51% mortality when they are exposed to LD50 dosage. And in contrary to that, there is no mortality is observed in channel and hybrid catfish. So this led to a question like, do the survivors acquire protection immunity against virus? Can blue catfish virus can protect against the ICHV1? So we would like to test the cross protection. So first, what we did is that, so there is 2 experiments, one to check the RFLP groups, another is blue catfish virus. So we just pooled all the survivors from the RFLP group A and RFLP groups B, and we selected just 1 strain from A and 1 strain B. And we did the same challenge model, everything same. So the survivors from A group and B group channels, they are re-exposed with ICHV1A. So as you can see, there is a significant cross protection effect has been provided by the previous exposure of the any of the RFLP group compared to the knife. But within the group, there is no significant difference between RFLP A group and B group. In the same way, again, the same survivors, that A group survivors, B group survivors of channels and hybrid, they were re-exposed with the ICHV1B. So similarly, so it does previous exposure to the any of the RFLP group viruses does give a protection, cross protection compared to the naive group. So again, as our third experiment, what we did is we took the BCAHV survivors, the channel, which survived the earlier blue catfish challenge, and hybrids, and we re-exposed that with the O-borne type strain. So as you can see, those who were exposed before did show 100% survival. So in order to summarize this, exposure to blue catfish alloherpes virus infers protective immunity in channel and hybrid catfish against ICHV1A. And so in my next objective, I would like to estimate the prevalence of the latency in the catfish hatcheries of Mississippi. So in order to do that, I collected like 200 fry per trough, and we selected 10 troughs of 6 hatcheries, and we did this sampling 3 times. And earlier report is that like the normal CCV latency is prevalence is around 10%. So Dr. Hansen has developed a qPCR assay to detect the active infection, which usually shows up at a CQ less than 32. Now, we would like to validate this, how this PCR is going to perform when we test the latency, which usually shows up after 33 cycles and which can go up to 40 cycles. So this is our protocol, and we used a 60-degree annealing temperature, and this is our master mix, and we have a common primers, forward and reverse, and we used two different probes, which is FARM for RFLP A group, Sci-Fi for RFLP B group, and we used SPUD as internal positive control. So when we perform the standards, so this looks fine, but you can see a cross reaction here. So RFLP A group does a cross reaction with B. So this is not supposed to be there. So we were thinking, what is the problem? So then we thought that it could be a contamination of standard or a sloppy bench work. So we made a new standards and new reagents, but again we have problems. Then we thought like, okay, there is only 2 nucleotide difference between the probe, that could be the reason. Again, so what we did is, instead of running a triplex or a duplex, we did individual PCR, like RFLP A group separate and B group separate. So it pretty much solved our problem. So again, but when we tried it with the hatchery fries, you can see almost all the samples are positive. So we would like to confirm that, hey, is it a real positive or is it a false positive? In order to do that, so there were some non-specific amplification at 60 degree, as we saw earlier. So what we did a gradient PCR, and at 64 degree, we don't have any non-specific amplification. So that solved our one problem. The second thing, as you can see, the CCO cells are consistently showing a positive amplification for RFLP A. For EB, there is no amplification, and there is no amplification for RFLP B with CCO cells and EB, and no template control is also negative. So we would like to know whether is it our contamination or is it anything. So we requested Dr. Sodo for some genomic DNA. So he has sent us tilapia, sturgeon, and koi DNA, and we have just extracted gill DNA in our lab. So as you can see, again, all others, tilapia, sturgeon, koi is negative, still we get a positive amplification for catfish genomic DNA. So at this point, we have new information, new catfish genome, and we have sequenced all the ICHV genomes. So we would like to, we designed a new primers and probes to test. So is it really a false positive, or is it actually a latent one? So we are proposing like, okay, less than 35 cycles. Okay, yeah, it's positive, 35, 37, suspect positive. And in 37 cycles, greater than 37 cycles, it will be a suspect negative. Now, coming into the genomics part, so this is our paper where we did the complete genome sequencing of blue catfish virus. So which is pretty similar, 93.9% similar to ICHV1, except it doesn't have OR of 16A. So if you look at this graph, you can see 99.2 and 98.6 similarity to the terminase gene as well as the polymerase gene, and compared to the ICHV1 and blue catfish virus. Now, I have a lot of genomes to analyze. So herpes virus genome is very complex and it is difficult to assemble. So there are multiple issues when we try to do the genomics. So the first issue is that we need to know some Python programming. Second issue is that we have to run all these packages one by one with a different data set, and we have to repeat it. That can have some problems. And there is another issue, is the installation of these packages can cause problems. This is a problem which I got with Guppy from Nanopore. Even they couldn't solve it. So this is a really difficult situation for a normal molecular biologist. So in that regard, I have developed a new package for NGS data. So in this, I'm using three things. Basically, SnakeMake, which is a program which automatically runs each package through all our samples. There is another package called Conda, which is basically installs all the program considering all the dependencies as an issue. And I've used Python to make a command line interface. So I can answer on this later in the section if anybody would like to know or would like to see a live demo. So so for my package requirements is just the Ubuntu, Conda, SnakeMake, and Python with minimum requirements. We can run it with an automatic pipeline without any programming background or Linux background. So coming to the potential project impacts, so information on variation and stability of the genome over the past years. So we are looking forward to for that. And so probably we would be able to identify a stable region by comparing all the isolate, which may help us to develop some chemotherapy. It may also help to identify potential vaccine target. We already got one. It may also help to identify virulence factors and latency factors. And this mechanism-based management strategies will reduce monetary losses for fish farmers and possibly allow for the expansion of the industry. So this is my team. Dr. Hansen is my major professor. Dr. Griffin is my co-major professor. My committee members are Dr. Weiss, Dr. Perkins, and Dr. Walsek. And we collaborate with Dr. Walbisa, Dr. Kuti Chandran, and Dr. Davison. And I got funding from India, Indian Council of Agricultural Research, and ORGS, CVM, Mississippi State University, Department of Basic Science, CVM, Mississippi State University, WFA Department, Mississippi State University, and of course, NMAT Thad Cochran, NMAT Center of Mississippi Delta. And this is our technical team. I would like to thank them too. These are some of my reference. And thank you. And I'm looking forward for the questions.
Great. Thank you very much, Aaron. That was another excellent talk. Feel free to unmute yourself, everybody, if you've got a question for him, or you can put it into the chat box and I can read it. Hopefully you can hear us, Aaron.
Hi, Aaron. This is Alex Primus from the University of Minnesota again. Can you hear me?
Yes.
Yeah, we can hear you, Alex. I think the problem's on his end.
Yeah.
All right. Let me.
Yeah. The problem is I cannot hear it.
All right. So I've opened up the chat box here.
Hey, Nick.
Yeah.
Arun's I'm going to have him just come down here real quick. The way they're set up, there's going to be
They can hear me, right?
feedback and all that. So, Arun, are you online now? Can you hear?
Alternatively, Matt, people can put the questions in the chat box and you can just answer them.
Okay.
Okay, he's running. Thanks for your patience, everybody.
And it's, this newfangled technology, Nick, it takes us a while to get caught up. We just got wireless installed last week.
Oh, well, welcome to the grid, Mississippi.
Here.
Hi, Aaron. Can you hear now?
I can hear you.
All right, mute yourself here, or whatever. Yeah.
Okay. Sorry for the technical thing.
No problem. So if you can hear me, this is Alex Primus from the University of Minnesota again. Very nice talk. Thank you for sharing.
Okay.
With respect to the protective effect of the alloherpes virus. I'm sorry, I may be mixing this up. The allovirus protective effect on re-challenge with the more traditional strains. Do you have any evidence after that initial allo challenge for the hybrids or the channel catfish that there was any replication of virus or persistence of virus between the time of that challenge and the re-challenge with the different virus?
Yeah, that's a good question. Basically, we were not planning to do this, but when we used the blue catfish virus, and after that, when we saw that, okay, all the channels and hybrids were survived, and we just checked it. So probably this year we are going to redo that, and we are planning for probably the virus replication, and we would like to check whether any about the antibodies are present. So yeah, we would like to investigate that further. But right now we don't have any data on that, but we would like to investigate that further.
Okay, yeah, I think that would complement things nicely. I guess I don't have too many concerns about it, but I guess it brings up the question of whether there may just be heightened innate immune activity going on at the second challenge. I would think it's unlikely, but if you looked at a few of those things, that might help.
Yeah. So especially when we look at the antibodies, and if we couple that with the viral replication assay, I hope we can answer that question, and it can give us good information.
Okay, thanks.
This is David Scarf. I did try and drop something into the chat box. Nick, do you want to ask the question, or should I?
You can go ahead and do that, Dave, if you want.
Yeah, thanks for an interesting presentation here. Do you have any information about the presence of any of these herpes viruses in either aquaculture or wild populations? And the second part of that is that given that all herpes viruses will become resident in spinal nerves of the host, and once the animal is stressed, it will then shed it. So they become lifelong reservoirs for this. Do you have any feeling for the epidemiological of this on any catfish, let's say in the US?
So you're asking about the wild fish, or?
Doesn't make any difference, wild or cultured.
Okay, so as far as the latency is concerned, in 2005, our lab has conducted the study. So we know that there is a 10% prevalence across different farms, and we do get outbreaks now and then. Last week in our diagnostic center, we got the case. So yeah, it's still out there. We do get the outbreaks, and we know that there is latency in the fries. So those fries are we are collecting from the trough before they get exposed to the wild or pond conditions. So those fries latency confirms that they transfer through vertical transmission, and we could see outbreak in the ponds also. Yeah, so latency is confirmed, and we do get the cases. So, but specifically, if we, for example, we get brooders with positive and breed them, and then grow them and see whether they express it, we didn't do it. But I would say circumstantial evidences from the multiple cases and multiple studies, we can say that, yeah, it's possible there is a vertical transmission and latency, and it can get reactivated. But we may have to perform a full experiment to conclusively say that.
That's what we're hoping, that it's pretty safe.
Okay, the second part of the question then is, given that all infected fish then become reservoirs, have you got any suggestions for how to control this disease and not allow it to spread, any of the herpes viruses?
So that probably I would say a vaccine, of course, can save it. But as you know, and everybody knows, it's pretty difficult. And herpes is not only a problem in fish, and you can see herpes simplex virus in humans. And after this many years, research and so much funding, still we are not able to success, because again the problem is with the herpes is so complex, and as we all know, the latency is another problem. So probably so if we ask a strategy for that, rather than going for the entire live organism, we can go for a like a spike protein, like what they did in Ebola. They used the vesicular stomatis virus and they put the spike protein outside that. So if you can get a particular protein and incorporate into a virus or any vector, so that means only the spike protein goes into the host, so they cannot cause latency. But if it's an active virus, yeah, it can cause a latency. So that's an issue. That probably, yeah, that can solve, but let's see.
Great, we got time for one more quick question. There's one out there.
So Arun, this is Tom Locke from Michigan State. I'll ask you a little bit more of a technical question. So we're currently playing around with an asepensor to herpes virus. So for your blue channel, or your blue channel catfish virus, was that it? The alloherpes virus. Do you have are there any special anything that you're doing special for a preservation standpoint in terms of cryopreservation, or you're just using your routine methods?
Oh, it's all same. What I do CCV, the same processing. So usually, if it's from the virus stock, if it's a virus stock, I just use a 30 microliter of virus stock and dilute with a 300 microliter of media without serum. Then just remove the media from the cell culture. So we use the CCO cell lines and just allow it to stay for 30 minutes and just roll it every 10 minutes. After that, add a fresh media with the 2x PS with antibiotics. So next day, within 14 or 16 hours, CP will come. So I just, then we can directly freeze it at -80. So when we take it out, we just do a 2 or 3 freeze-thaw cycle, freeze-thaw cycle, so that that can release the viruses. So that's pretty much I do same for CCV and blue catfish, this blue catfish alloherpes virus. So they're pretty much same.
Excellent, thank you.
All right, thanks again, Aaron, for the great talk. Round of applause to you. And thanks again. Just reminding everybody that next week we will not have presentations, but we'll be back on July 10th for 2 more at the same time, and you can watch your email or the website for the Zoom link to join us. Have a great weekend, everybody.